Hello, everyone. Welcome back. This is Dr. Tarun again. We are now in segment two of Unit 3. And in this segment, we will be covering chapter 9 of your textbook on cell signaling. We will not go through the entire chapter, however. It's going to be a very truncated description or discussion of chapter 9. We will only cover sections 9.1 through 9.3. So please take note of that. We won't cover the two final sections at all, which is 9.4 and 9.5. Before we enter into the subject, I would like to make a provocative or even perhaps a controversial statement at the start, not just to grab your attention, but to get you thinking more deeply about our present topic, present phenomenon that we are going to learn. And I will say so within the context of your liberal college education here at the university. So, part of your liberal education here is to push the limits of your perception about nature and its inner workings. In doing so, we as educators, a key part of our mission, would like to train you to be able to view phenomena with multiple sets of intellectual lenses so that your mind will be set free to think outside the box, which is always a fresh way of looking at things and be creative, allowing you to formulate questions in your mind with fresh sets of intellectual eyes and alternative points of views. And this is critical because you will be the next generation of thinkers, movers, scientists, educators, policymakers, civil servants. In other words, you will be the ones who will push the borders of science, technology, and human progress in our society and even in the world. So you have that potential and promise. So, in light of that, here is the controversial statement that I wish to say. You don't need a brain to be intelligent. Let me say that again. You don't need a brain to be intelligent. And I hope to convince you at the end of this chapter of that statement, and in fact, the next chapter, chapter 11 on cell division and cell cycle, that there is indeed a profound truth to this statement. To start with, for example, the fundamental essence of intelligence is the ability and the capacity for complex communication involving three basic elements. First, the perception of a signal from a source, asking or answering the question, what is it? Two, the decoding of the perceived signal, which answers the question, what does it mean? And third, the execution of an appropriate response to the perceived and interpreted signal, or answering the question, what should I do? Or what is my reaction? Remarkably, the microscopic yet highly complex chemical factory called cells that we came to know in chapter 5 also has these incredible attributes. The cell does not, or does so in the most exquisite way, beyond what even the most modern, brilliant human engineers and computer scientists can build in all their technological capabilities. And to be sure, we're not, of course, talking about psychological intelligence or the intelligence of the mind, although it is a foundation, in fact, for the emergence of it from a single cell. Because you consider the fact that you, a beautiful organism made up of 37 trillion cells, carrying a rational brain made up of at least 100 billion cells. We all started from a single egg cell. Now, how did we come about? A major reason is that the cell possesses an extremely complex and elaborate form of what we call as chemical intelligence. Chemical intelligence. To a computer engineer working on nanomachines and artificial intelligence, the living cell, in fact, is the most computational machine with nanoparts, bathed and operating remarkably well, in fact, in water. So it is a computational machine that is not just capable of reacting to its immediate environment, but is able to transform itself into the most complex system that we now call life. And not just life, but intelligent life. So, with that long interlude, I would like us to start now our journey into this chapter on cell signaling. Your first reading assignment is, of course, section 9.1 on the principles of signaling, cell signaling, which covers page 185 to 188 of your textbook. So here are the learning objectives of 9.1. So the major objective or the main objective is to predict how changes in the chemical signaling pathway may affect cell communication. And the sub-objectives in that is that you should be able to describe the four essential elements of cellular communication. And you should also be able to describe the process of signal transduction from signal molecule to receptor to amplification and finally to termination. After reading section 9.1 and watching this video, try your best to answer these following self-assessment questions. One, what are the four essential elements of cell signaling? Two, what are the steps that occur when a signaling molecule binds to a receptor on a responding cell? And three, if a hormone is released into the bloodstream so that it comes in contact with many cells, what determines which cells in the body respond to the hormone? There are four core concepts or principles that underlie cell signaling that you need to learn in this chapter. First is cells can communicate over long and short distances. Second, signaling molecules bind to and activate cell surface and intracellular receptors. Third, G protein coupled receptors are examples of a large conserved family of receptors that often lead to short term responses. And fourth, receptor kinases are widespread and often lead to long term responses. Figure 9.1 in your textbook highlights these essential elements for communication. The first is the signaling cell, the signaling molecule, the receptor molecule, and the receptor cells. So these are the essential elements for cell to cell communication. So the signaling cell, as depicted here, is responsible for releasing the signaling molecules via what we call as the secretory pathway. And the signaling molecule then diffuses across a distance until it contacts a specific structure on the surface of another cell. This cell that receives the signal is called the responding cell. And the specific molecule that is found on the cell that receives the specific signal is called the receptor protein. So this is, in essence, what is happening in all forms of cell-to-cell communication. So there is the signaling cell, the signaling molecule, the receptor molecule, and the receptor cell. These four essential elements of cell-to-cell communication is universal. It is both found in prokaryotes and in eukaryotes. Now, in the case of a prokaryote like bacterial cell, a classic example of this is, and it's shown in your textbook, is the uptake of DNA from the environment in Streptococcus pneumoniae, for example, where this is first observed. it was observed that this uptake increased when the population of the bacteria reached a certain density. In the 1990s, a peptide consisting of 17 amino acids was discovered to be continuously synthesized by pneumococcal cells. And the receptor for this peptide was discovered to be found on the surface of the cells. So in this figure, the signaling cell is actually the 17 amino acids peptide that was discovered in the 1960s, and subsequently they discovered a specific receptor protein for it on the surface of the bacterial cell. Now, when the peptide binds the receptor, the bacterium actually expresses specific type of genes that enable it to take up DNA from the environment. Now, if a low density of bacteria is present, this peptide is made at low levels, with the result that the genes for taking up the environmental DNA are also low. However, if a high density of the bacteria is present, the peptide is made at high levels, and the result is environmental DNA uptake. So based on studies like this in bacteria, as well as in studies in eukaryotes, have actually uncovered 4 basic steps in cell signaling. Okay, of course, including the elements of the first events, which is the cell-to-cell communication. So the 4 basic steps in cell signaling are receptor activation, signal transduction, the specific response or set of responses, and then the termination of the signal. Now, once the signaling molecule is bound to the receptor, like so, so this is the signal binding to the receptor, which is that portion of the responding cell. The receptor is turned on, or we call it activated. And once this activation process has occurred, then it transmits the message into the cell through the cytoplasm in a process called signal transduction. And this signal transduction pathway consists of multiple elements of typically different specific types of proteins as well. Okay, and this message through the signal transduction can either remain in the cytosol in both eukaryotes and prokaryotes, or in the case of eukaryotes, it can go all the way to the nucleus. So the signal transduction pathway can extend beyond the cytoplasm or the cytosol, but it can also cross the nuclear membrane and proceed to the nucleus in the case of eukaryotes. So if it is in the cytosol, usually it elicits a rapid or short-term response. And this happens by, for example, activating an enzyme, or simply allowing certain events that happen in the cytoplasm, like regulating the endomembrane system, for example, in secreting protein or secreting molecules. Okay, so in eukaryotes, in terms of extending the signal transduction pathway all the way to the nucleus, usually this triggers a slow but long-term response. and this usually involve turning on of the genes. Okay, and when it involves turning on of the genes, usually what happens here is that you cause other long-term responses such as cell division. Soon understand its details in chapter 11. So there are multiple types of responses as a result of a simple binding event on the surface of the cell during the receptor activation process. So the main important step here is the nature of the signal transduction pathway, okay, which determines the type of response that the cell will have. And of course, it is always important to have the final step called the termination, because we need to get the process stopped when the cell no longer needs to respond to the signal. So these are the four basic steps in cell signaling. So now going back to our original example of the bacterial cell, the pneumococcus example. So the peptide signaling molecule shown here binds to the receptor on the cell surface. When enough peptide is bound, a signal is relayed by signal transduction pathway, not shown in this diagram, but which now you can understand is what is happening. And it actually binds to the nucleoid region. Remember that the bacterial cell does not have a nucleus, but the DNA is found in a region called the nucleoid. Okay, okay, where the DNA is, and then the genes, the specific genes are turned on that expresses certain sets of proteins involved in DNA uptake from the environment. And so this happens at high population density. At high population density, the concentration of the signaling peptide is high enough in order to bind to the receptors sufficiently to stimulate DNA uptake as a result of turning on of genes. And these are all relayed by the signal transduction pathway inside the bacteria in its cytosol. So what happens with cell signaling at low bacterial cell density? So at low population density, the concentration of the signaling peptide, as shown here, is too low to bind to the receptors. And therefore, the receptor is unbound and therefore is not activated. And therefore, the signaling transduction pathway is also not relaying the signal. And therefore, there is no transcription of genes that is responsible for DNA uptake. And therefore, there is no stimulation of DNA uptake. So this is how the signal transduction mechanisms at low population density can be explained. So this is the end of section 9.1. But before you finish this video, I would like you to answer this RIF question. So please open your RIF accounts and try to answer this following question, which is designed to allow you to test your understanding of objectives 9.1a and 9.1b. So I'll see you in the next section. Thank you.